Welcome to the tour of a Smart Science College Biology Lab. Today we're going to review the Smart Science lab called mitosis. I want you to notice a few things as we go through this Smart Science tour. Number one is Smart Science is the only online lab to use real interactive video for all of the interactive elements within the lab. Nothing is animated or simulated in a Smart Science lab. And number two is our use of the scientific method. And so first I want to point to this bar at the top where the students navigate through Smart Science. You notice that we are very centered around students making predictions and formulating hypotheses, going into a lab to try to gather evidence to support these hypotheses, and then writing up their findings in a lab report at the end. And in this way we have embodied the scientific method very strongly within our system. So here's our first page at the beginning of a Smart Science lab tour. It's called the Think page. The Think page contains a basic lab description with some background materials to try to get the students warmed up on the topic. In this case, we're talking about the cell cycle, we're talking about mitosis, and how the students will interact with our slides and images within this lab. Then they go on and answer some warm-up questions. These can be customized by a professor to fit the needs of your class. So don't feel that you only have what sets we have available to you. We can put in your questions as well. Once the student has gotten through answering these different warm-up questions, they will hit the Submit Answer button. This will grade the quiz automatically as well as give the students some feedback on how to get the answers right in the future. And now we're going to move on to the second page of Smart Science, the Predict page. Here the student will get a basic audio and video background for the lab. So here in the audio they're explaining what mitosis is and what you're going to look for when you're doing this particular lab, what the slides consist of. At this point we ask the students to try to formulate a hypothesis and find something they'd like to test out within this lab. Now they may write their own hypothesis or they may pick from a list. Here I'm showing the write your hypothesis option. So I'm going to write a hypothesis in the cell cycle. Most time is spent in metaphase. Let's go ahead and press the Enter key, add this hypothesis to my list, and now we can go ahead and try and test this hypothesis in our lab. I'm going to press the Enter Lab button to get started. Okay, now we're within the Smart Science lab, and the basic components here are at the top, my hypothesis. Below that, I can pick from a number of different cell types that I want to look at, and then I can watch and measure from each of those different video and image combinations. I'm just going to go ahead and pick out the simplest cell type to start with, a very well magnified stem cell setup. Okay, and here we have our stem cell slide. I'm going to go ahead and press this watch button. It's going to show a basic setup for this lab, a bit of a procedural element. We see that a slide is being set onto a microscope, and then the focus is adjusted, at which point we now adjust to a more magnified eyepiece, and then the fine focus is adjusted, and then we see those stem cells. So you do get a bit of information within that video before you start measuring. At this point the measure button is lit up, and this is really the heart and soul of Smart Science. The ability to measure some different facet of this lab. In our case, I'm going to be marking cells that I see that fit different phases. For example, here it says prophase in orange, and if I mark a cell, it's going to put an orange circle on that cell. It's going to put a number in the database next to prophase, and it's going to put a bar graph on my graph with the word prophase. Okay, I think I see one other that might be a prophase, and then I'm going to move on. Now metaphase. I'm quite good at finding metaphase. The chromosomes are lined up in the middle, so like these two right here, with a light green marking that I've created, and that looks like just about the only metaphase. Now I'm going to go on and do anaphase. 1, 2, 3, and 4. And you can see the anaphase over here on the graph. Now I will go ahead and do telophase. I don't see a whole lot of those. There's a possibility for one of these two, but I'm uncertain, so I'm just going to skip on. Lastly, interphase. And this is any time that I can clearly see a circle nucleus within the cell. My understanding is that many of these cells look like interphase, and we will see that noted on the graph to the right. In fact, the bar for interphase is quite a lot higher than the other bars that I've been creating. And you can see that I've marked many cells as being clearly interphase. I'll now press the Done button, and I'll look over at my graph and notice that this does not well support the hypothesis I've written. In the cell cycle, most time is spent in metaphase, I thought, but it seems that the most cells are in interphase, so I've found some evidence to contradict what I've written. Alternatively, we could go back and look at some other cells, such as these other onion root types. Maybe this one would be quite nice. Collect some more data and see if that matches or contradicts the data that I've already collected. In this way, smart science is a nice iterative process of allowing students to refine their hypothesis, collect some data, see if it impacts their thinking about that, and move on. Okay, more than just doing the data collection, now we have the ability to review some vocabulary words on this page as well. And you'll see all these different words down at the bottom in blue buttons. Say that I'm not really sure what binary fission is. I can click on that word, and binary fission comes up. There it is. Reproduction of a cell organelle by simple division into approximate equal parts. There are many words for each Smart Science lab the student can research. But at this point, they are able to take a quiz and try to express what they've learned in the form of a summative assessment. These questions again can be customized by the instructor, so don't feel that you have to use our questions, although we do have several difficulty levels of question available already. Once I've gone ahead and answered each of these post-lab questions and submitted my answers, a new button appears in the lower right-hand corner that says Write Report. This is really one of the big strengths of Smart Science, as many other online labs do not have nearly as good of a lab report as we do, or even have one at all. We feel the strength of doing a lab work is expressing your findings to the instructor, and here you get to do it in an open format with some prompts and bullet points to help the student as to what to write. Again, you can customize what prompts and what bullet points your students see, and even how many written boxes they have. This is our basic default format. I can write this lab is about cells, and some more. You really have as much space as you'd like to fill with writing, so the student should not feel as if there's a limit on how much they can write in each of these areas. They get a procedure section, and then we show the data that the student collected in 3 formats: a data table, a graph, and a picture of the experiment they were working on, with all the exact clicks that I made. So you as an instructor, when you get this reported back to you, will see exactly which cell they chose for what type. That is very powerful indeed. We then ask the students to write some results, review their hypothesis, and try to make a conclusion based on that hypothesis that they created and all the data they've now reviewed. At this point, they can submit their report for grading, and I will do so. And now the Smart Science Lab has been completed. There is some extra credit available, but I'm just going to hit this Leave Lab button and leave you here with a finished tour of a Smart Science Lab about mitosis. Thank you so much for your time today, and I hope you enjoyed looking at one of our labs.